Hello everyone, welcome back to my channel. After a really long time, here I am with a new topic, and today's topic is from Unit 4, and the name of the subtopic is Methods of Physiological Psychology. So before going into the topic, let's see what physiological psychology means. So it is also known as biological psychology or biopsychology, and it's the study of the physiological basis of behavior. And when we say physiological basis, the basis is our brain, and we will study how our brain processes affect our behavior and feelings. So this is the third point, which says that it aims to understand how our brain functions to control our behaviors and feelings. So in order to study the brain functions, there are various techniques, and this unit is this subtopic from this unit is about those techniques. And what are the methods? The methods are divided into two types. One type is known as the invasive methods, and it involves the use of instruments to cut skin or other connective tissue, or the insertion of instruments into a body opening. So invasive methods means where one has to cut skin or the connective tissue, or one has to insert instruments into a body opening. So those are invasive methods. There are non-invasive methods too. Here let's see what are some of the examples of invasive methods in medical science. Examples are biopsy, endoscopy, etc. And in our syllabus for UGC NET psychology, we have these methods prescribed in the unit, and these are anatomical methods, degeneration techniques, lesion techniques, chemical methods, microelectrode studies. Then another type is the non-invasive methods, and in non-invasive methods what happens is that this does not involve break in skin or contact with internal body cavity. So it's the opposite of the invasive methods. Here no cut in the skin is involved. So that's why it is called non-invasive method. And one example from the medical field can be ultrasonography. And in our syllabus for biological psychology, the methods prescribed are EEG and scanning methods. And today's topic is all about a brief introduction to each invasive method. So today we won't go into details of the non-invasive methods. Today we will just touch upon the invasive methods prescribed in our syllabus, that too only a brief introduction of each, and we will go into the details of each topic one by one in our later videos. So first let's study about the anatomical methods under invasive methods. So what are the anatomical methods? Methods used to study the structure of a part of the body, and here brain in case of physiological psychology. So we study the anatomy or the structure of a part of the body, and that's why these are called anatomical methods. And for biopsychology, the brain structure is studied. And what are some of the methods used under anatomical methods? CT scan, MRI, fMRI, PET scan, and near infrared imaging. And there are other such anatomical methods, and these fall under invasive methods, though these are minimally invasive methods. So no drastic cut in the skin or break in the skin or insertion into body cavity is involved, but there are minimal invasions going on. For example, for MRI, a person is made to drink a particular liquid, a chemical, which makes the image clear when the person undergoes the scanning. So also there are involvement of rays, X-rays for that matter. That's why these are invasive methods, though minimally invasive methods. Then we have degeneration techniques, and here let me take the opportunity to share this with you that I could not find proper and adequate resources for this particular topic, which is degeneration technique, especially for biopsychology. There are other topics for medical science topics in the internet resources, but for biopsychology I could hardly find any article or videos for that matter, and it was a bit difficult to note down this point, though a very simple statement, but still in order to understand and then prepare this slide for degeneration technique was tedious for me due to lack of resources. Anyways, so what are degeneration techniques? These techniques are used to trace neural connections in experimental animals by cutting off a nerve fiber. So neural connections are traced with the help of these techniques, and what is done in the degeneration techniques? A nerve fiber is cut off. And there are two types of such techniques, or I should correct the statement that there are two types of degeneration, and one type is the anterograde degeneration, and under anterograde degeneration a pattern of neuron destruction following axonal injury that spreads forward along the axon away from the nerve cell body, also called Wallerian degeneration. So in this particular degeneration there is an injury in the axon, and the injury spreads forward along the axon and it is moving away from the nerve cell body. And there is another name for anterograde degeneration which is called Wallerian degeneration. Another type is the retrograde degeneration, and this is the reverse of anterograde degeneration. Here a pattern of neuron destruction following axonal injury that spreads backward along the axon toward and then into the nerve cell body. So the effect of injury does not spread forward, rather it spreads backward along the axon and moves toward the nerve cell body. So this is the difference between anterograde degeneration and retrograde degeneration. Now let's move to the lesion techniques. What are lesion techniques? Lesion means a damage to a part of the brain, and this can happen due to a brain injury or a disease, and sometimes these are deliberately created in experimental animals in order to study the effect of that particular lesion on the behavior of the animal. And the function of a brain area is inferred by observing the deficits that are produced when the area is removed from the brain. So this is all about the lesion techniques. For this particular discussion in this video, we will go into the proper details, in-depth details in our next videos, later videos on each of the techniques. So I will have, for example, a proper video only on lesion techniques. So today it's just the brief introduction of each of the invasive techniques. Then comes the chemical methods, and in a chemical method what happens is that a cannula or a tube is used to place chemicals in parts of the brain that can kill or influence neurons, and chemicals are inserted. And when we study chemical methods, we will come across two terms, agonists and antagonists. What are agonists? Agonists are those chemicals or drugs which increases neurotransmitter effects. Okay, and what are antagonists? Antagonists are those chemicals or drugs which decreases neurotransmitter effects. Another topic is the microelectrode studies, and in intracellular, microelectrodes are used to stimulate and record neural activity within a single neuron. So what happens in microelectrode studies? Microelectrodes are used, okay, these are intracellular, and intracellular means in one cell, and These are used to stimulate and record neural activity within a single neuron. Okay, as I have mentioned, in a single neuron, the microelectrodes are inserted to record neural activity. And the tip of the microelectrodes are with tips less than 1 microns in diameter. So the diameter is less than 1 microns most of the time. So that's why they are called microelectrodes. And they function as an electrical conductor transmitting the electrical information in the brain. So microelectrodes, they work as electrical conductors transmitting the electrical information in the brain. For today's video, this brief introduction is enough. We will go into the details again in our later discussion in later videos. And thank you for watching. I have started, and hopefully I will put regular videos in my channel from now onwards, and that's why please consider liking, sharing and subscribing to the channel. Like the content, share it with your friends who are in need for guidance for UGC NET examination and other examinations which involves psychology as a subject. And that's how. Please be connected. Thank you very much. We'll meet again. Bye bye.